Dual virus origin theory. Did a cell emerge from the fusion of a genetic virus and an energetic virus? Abstract. Existing hypotheses on the origin of life include the RNA World Hypothesis, viral origin theory, and endosymbiotic theory. This paper proposes a new scenario in which a genetic virus and an energy-producing virus, similar to mitochondria, fused to form the first self-replicating cell. This theory offers a reinterpretation of viruses' evolutionary roles and the structural emergence of cells, inviting philosophical discussions on the definition of life itself. 1. Introduction. The question of how life began spans biology, chemistry, and philosophy. Current theories, such as the RNA World Hypothesis, Endosymbiotic Theory, and Viral Origin Theory, attempt to explain the rise of cellular complexity. However, each offers partial insights and lacks a complete explanation of cellular genesis. This paper introduces a novel perspective. A cell may have originated from the fusion of two distinct viruses, one carrying genetic material and the other capable of energy production. This union suggests that life's foundational elements could have arisen not from independent units or competition, but from cooperation and symbiosis. 2. Related hypotheses and theoretical background. 2.1 RNA World Hypothesis posits that early life was based on RNA, which can store genetic information and act as an enzyme. It provides a framework for understanding self-replicating systems before DNA and protein evolution. 2.2 DNA Viral Origin Theory. Researchers such as Patrick Forterre have proposed that DNA viruses introduced DNA into RNA-based organisms, enhancing genetic stability and shaping evolutionary pathways. 2.3 Endosymbiotic Theory. Lynn Margulis suggested that mitochondria and chloroplasts originated as free-living bacteria that became part of eukaryotic cells through symbiosis, providing energy and metabolic functions. 2.4 Evolutionary Role of Viruses. Modern studies suggest viruses are not merely pathogens but evolutionary agents that transport genetic material and reshape host genomes, supporting the possibility of a viral fusion origin for cells. 3. Proposed theory. Dual virus fusion scenario. This paper presents a hypothesis in which two viruses, one with genetic replication ability and one with energy production functionality, fused to form the first cell. Their collaboration could have enabled energy metabolism and genetic control within a single entity, leading to the characteristics of modern cells. 3.1 Role of the genetic virus. Likely RNA or DNA-based, such viruses replicated genetic information and responded to environmental stimuli, driving reproduction and adaptation. 3.2 Function of the energetic virus. Hypothetically possessed internal structures or enzymes capable of producing chemical energy, akin to proto-mitochondria, allowing survival without a host. 3.3 Fusion mechanism. Contact and integration. Energetic virus binds or penetrates genetic virus. Functional synergy. Genetic virus controls or utilizes the energy metabolism of its partner. Evolution into single entity. Formation of stable membrane-bound system with internal compartments. Acquisition of cell-like properties, replication, metabolism, and environmental responsiveness emerge. 3.4 Transition into cellular life. This fusion structure, combining energy and information systems, evolves into a self-replicating unit capable of adapting and propagating, marking the birth of true cellular life. Comparison with existing theories and implications. 4.1 Differences from endosymbiotic theory. While endosymbiotic theory involves bacteria and archaea forming eukaryotic cells, the dual virus origin suggests mitochondria-like structures were viral, not bacterial in origin. 4.2 New interpretation of RNA-DNA transition. The transition from RNA to DNA is here seen as a result of viral intervention. DNA was introduced, not just naturally evolved. 4.3 Redefining virus cell boundaries. This hypothesis blurs the line between viruses and cells, suggesting cells may be highly evolved viral structures, prompting philosophical reconsideration of what constitutes life. 4.4 Cooperative model of life's emergence. Rather than competition and survival, life's origin may have been rooted in cooperation and the merging of complementary functions, offering a more holistic view of evolutionary beginnings. Conclusion and future research directions. This paper proposes a dual virus fusion theory for the origin of the cell, integrating elements of genetic control and metabolic energy into a single living unit. It repositions viruses not as mere disease agents, but as evolutionary innovators and building blocks of life. 5.1 Significance of the theory. reframes viruses as active drivers of biological evolution, presents a fusion-based mechanism for cellular genesis, invites philosophical discussions on defining life. 5.2 Future research possibilities. Identify modern viruses with energy-producing traits. Compare mitochondrial evolution with viral systems. Model genetic integration between viruses in early life. Conduct biochemical simulations of viral fusion.